Chapter 42 The Immune System Figure 42.1 In this compound light micrograph purple-stained neutrophil, upper left, and eosinophil, lower right, are white blood cells that float among red blood cells in this blood smear. Neutrophils provide an early, rapid, and nonspecific defense against invading pathogens. Eosinophils play a variety of roles in the immune response. Red blood cells are about 7 to 8 micrometers in diameter, and a neutrophil is about 10 to 12 micrometers. Credit. Modification of work by Dr. David Saba. Introduction. The environment consists of numerous pathogens, which are agents, usually microorganisms, that cause diseases in their hosts. A host is the organism that is invaded and often harmed by a pathogen. Pathogens include bacteria, protists, fungi and other infectious organisms. We are constantly exposed to pathogens in food and water, on surfaces, and in the air. Mammalian immune systems evolved for protection from such pathogens. They are composed of an extremely diverse array of specialized cells and soluble molecules that coordinate a rapid and flexible defense system capable of providing protection from a majority of these disease agents. Components of the immune system constantly search the body for signs of pathogens. When pathogens are found, immune factors are mobilized to the site of an infection. The immune factors identify the nature of the pathogen, strengthen the corresponding cells and molecules to combat it efficiently, and then halt the immune response after the infection is cleared to avoid unnecessary host cell damage. The immune system can remember pathogens to which it has been exposed to create a more efficient response upon re-exposure. This memory can last several decades. Features of the immune system, such as pathogen identification, specific response, amplification, retreat, and remembrance are essential for survival against pathogens. The immune response can be classified as either innate or active. The innate immune response is always present and attempts to defend against all pathogens rather than focusing on specific ones. Conversely, the adaptive immune response stores information about past infections and mounts pathogen-specific defenses. Chapter Outline 42.1 Innate Immune Response 42.2 Adaptive Immune Response 42.3 Antibodies 42.4 Disruptions in the Immune System 42.1 Innate Immune Response. By the end of this section, you will be able to do the following. Describe physical and chemical immune barriers. Explain immediate and induced innate immune responses. Discuss natural killer cells. Describe major histocompatibility class I molecules. Summarize how the proteins in a complement system function to destroy extracellular pathogens. The immune system comprises both innate and adaptive immune responses. Innate immunity occurs naturally because of genetic factors or physiology. It is not induced by infection or vaccination but works to reduce the workload for the adaptive immune response. Both the innate and adaptive levels of the immune response involve secreted proteins, receptor-mediated signaling, and intricate cell-to-cell communication. The innate immune system developed early in animal evolution, roughly a billion years ago, as an essential response to infection. Innate immunity has a limited number of specific targets. Any pathogenic threat triggers a consistent sequence of events that can identify the type of pathogen and either clear the infection independently or mobilize a highly specialized adaptive immune response. For example, tears and mucus secretions contain microbicidal factors. Physical and chemical barriers. Before any immune factors are triggered, the skin functions as a continuous, impassable barrier to potentially infectious pathogens. Pathogens are killed or inactivated on the skin by desiccation, drying out, and by the skin's acidity. In addition, beneficial microorganisms that coexist on the skin compete with invading pathogens, preventing infection. Regions of the body that are not protected by skin, such as the eyes and mucous membranes, have alternative methods of defense, such as tears and mucus secretions that trap and rinse away pathogens, and cilia in the nasal passages and respiratory tract that push the mucus with the pathogens out of the body. Throughout the body are other defenses, such as the low pH of the stomach, which inhibits the growth of pathogens, blood proteins that bind and disrupt bacterial cell membranes, and the process of urination, which flushes pathogens from the urinary tract. Despite these barriers, pathogens may enter the body through skin abrasions or punctures, or by collecting on mucosal surfaces in large numbers that overcome the mucus or cilia. Some pathogens have evolved specific mechanisms that allow them to overcome physical and chemical barriers. When pathogens do enter the body, the innate immune system responds with inflammation, pathogen engulfment, and secretion of immune factors and proteins. Pathogen recognition and infection may be intracellular or extracellular, depending on the pathogen. All viruses infect cells and replicate within those cells, intracellularly, whereas bacteria and other parasites may replicate intracellularly or extracellularly, depending on the species. The innate immune system must respond accordingly, by identifying the extracellular pathogen and/or by identifying host cells that have already been infected. When a pathogen enters the body, cells in the blood and lymph detect the specific pathogen-associated molecular patterns, PAMPs, on the pathogen surface. PAMPs are carbohydrate, polypeptide, and nucleic acid signatures that are expressed by viruses, bacteria, and parasites but which differ from molecules on host cells. The immune system has specific cells, described in Figure 42.2 and shown in Figure 42.3, with receptors that recognize these PAMPs. A macrophage is a large phagocytic cell that engulfs foreign particles and pathogens. Macrophages recognize PAMPs via complementary pattern recognition receptors, PRRs. PRRs are molecules on macrophages and dendritic cells which are in contact with the external environment. A monocyte is a type of white blood cell that circulates in the blood and lymph and differentiates into macrophages after it moves into infected tissue. Dendritic cells bind molecular signatures of pathogens and promote pathogen engulfment and destruction. Toll-like receptors, TLRs, are a type of PRR that recognizes molecules that are shared by pathogens but distinguishable from host molecules. TLRs are present in invertebrates as well as vertebrates, and appear to be one of the most ancient components of 1194 Chapter 42. The Immune System Access for free at OpenStax.org. The immune system. TLRs have also been identified in the mammalian nervous system. Figure 42.2. The characteristics and location of cells involved in the innate immune system are described. Credit. Modification of work by NIH, 42.1. Innate immune response 1195. Figure 42.3. Cells of the blood include 1. Monocytes, 2. Lymphocytes, 3. Neutrophils, 4. Red blood cells, and 5. Platelets. Note the very similar morphologies of the leukocytes, 1, 2, 3. Credit. Modification of work by Bruce Wetzel, Harry Schaefer, NCI. Scale bar data from Matt Russell. Cytokine release effect. The binding of PRRs with PAMPs triggers the release of cytokines, which signal that a pathogen is present and needs to be destroyed along with any infected cells. A cytokine is a chemical messenger that regulates cell differentiation, form and function, proliferation, production, and gene expression to affect immune responses. At least 40 types of cytokines exist in humans that differ in terms of the cell type that produces them, the cell type that responds to them, and the changes they produce. One type of cytokine, interferon, is illustrated in Figure 42.4. One subclass of cytokines is the interleukin, IL, so named because they mediate interactions between leukocytes, white blood cells. Interleukins are involved in bridging the innate and adaptive immune responses. In addition to being released from cells after PAMP recognition, cytokines are released by the infected cells which bind to nearby uninfected cells and induce those cells to release cytokines, which results in a cytokine burst. A second class of early-acting cytokines is interferons, which are released by infected cells as a warning to nearby uninfected cells. One of the functions of an interferon is to inhibit viral replication. They also have other important functions, such as tumor surveillance. Interferons work by signaling neighboring uninfected cells to destroy RNA and reduce protein synthesis, signaling neighboring infected cells to undergo apoptosis, programmed cell death, and activating immune cells. In response to interferons, uninfected cells alter their gene expression, which increases the cell's resistance to infection. One effect of interferon-induced gene expression is a sharply reduced cellular protein synthesis. Virally infected cells produce more viruses by synthesizing large quantities of viral proteins. Thus, by reducing protein synthesis, a cell becomes resistant to viral infection. 1196 Chapter 42. The Immune System Access for free at OpenStax.org. Figure 42.4. Interferons are cytokines that are released by a cell infected with a virus. Response of neighboring cells to interferon helps stem the infection. Phagocytosis and inflammation. The first cytokines to be produced are pro-inflammatory. That is, they encourage inflammation, the localized redness, swelling, heat, and pain that result from the movement of leukocytes and fluid through increasingly permeable capillaries to a site of infection. The population of leukocytes that arrives at an infection site depends on the nature of the infecting pathogen. Both macrophages and dendritic cells engulf pathogens and cellular debris through phagocytosis. A neutrophil is also a phagocytic leukocyte that engulfs and digests pathogens. Neutrophils, shown in Figure 42.3, are the most abundant leukocytes of the immune system. Neutrophils have a nucleus with 2 to 5 lobes, and they contain organelles, called lysosomes, that digest engulfed pathogens. An eosinophil is a leukocyte that works with other eosinophils to surround a parasite. It is involved in the allergic response and in protection against helminths, parasitic worms. Neutrophils and eosinophils are particularly important leukocytes that engulf large pathogens, such as bacteria and fungi. A mast cell is a leukocyte that produces inflammatory molecules, such as histamine, in response to large pathogens. A basophil is a leukocyte that, like a neutrophil, releases chemicals to stimulate the inflammatory response as illustrated in Figure 42.5. Basophils are also involved in allergy and hypersensitivity responses and induce specific types of inflammatory responses. Eosinophils and basophils produce additional inflammatory mediators to recruit more leukocytes. A hypersensitive immune response to harmless antigens, such as in pollen, often involves the release of histamine by basophils and mast cells. Figure 42.5. In response to a cut, mast cells secrete histamines that cause nearby capillaries to dilate. Neutrophils and monocytes leave the capillaries. Monocytes mature into macrophages. Neutrophils, dendritic cells, and macrophages release chemicals to stimulate the inflammatory response. Neutrophils and macrophages also consume invading bacteria by phagocytosis. Cytokines also send feedback to cells of the nervous system to bring about the overall symptoms of feeling sick, which include 42.1, innate immune response, 1197 lethargy, muscle pain, and nausea. These effects may have evolved because the symptoms encourage the individual to rest and prevent the spreading of the infection to others. Cytokines also increase the core body temperature, causing a fever, which causes the liver to withhold iron from the blood. Without iron, certain pathogens, such as some bacteria, are unable to replicate. This is called nutritional immunity. Link to learning. Watch this 23-second stop-motion video, http://openstax.org/l/canidia, showing a neutrophil that searches for and engulfs fungus spores during an elapsed time of about 79 minutes. Natural killer cells. Lymphocytes are leukocytes that are histologically identifiable by their large, darkly staining nuclei. They are small cells with very little cytoplasm, as shown in Figure 42.6. Infected cells are identified and destroyed by natural killer NK cells, lymphocytes that can kill cells infected with viruses or tumor cells, abnormal cells that uncontrollably divide and invade other tissue. T cells and B cells of the adaptive immune system also are classified as lymphocytes. T cells are lymphocytes that mature in the thymus gland, and B cells are lymphocytes that mature in the bone marrow. NK cells identify intracellular infections, especially from viruses, by the altered expression of major histocompatibility class (MHC) I molecules on the surface of infected cells. MHC I molecules are proteins on the surfaces of all nucleated cells. Thus they are scarce on red blood cells and platelets, which are non-nucleated. The function of MHC I molecules is to display fragments of proteins from the infectious agents within the cell to T cells. Healthy cells will be ignored, while non-self or foreign proteins will be attacked by the immune system. MHC2 molecules are found mainly on cells containing antigens, non-self proteins, and on lymphocytes. MHC2 molecules interact with helper T cells to trigger the appropriate immune response, which may include the inflammatory response. Figure 42.6. Lymphocytes, such as NK cells, are characterized by their large nuclei that actively absorb right stain and therefore appear dark-colored under a microscope. An infected cell, or a tumor cell, is usually incapable of synthesizing and displaying MHC I molecules appropriately. The metabolic resources of cells infected by some viruses produce proteins that interfere with MHCI processing and/or trafficking to the cell surface. The reduced MHCI on host cells varies from virus to virus and results from active inhibitors being produced by the viruses. This process can deplete host MHCI molecules on the cell surface, which NK cells detect as unhealthy or abnormal, while searching for cellular MHCI molecules. Similarly, the dramatically altered gene expression of tumor cells leads to expression of extremely deformed or absent MHCI molecules that also signal unhealthy or abnormal. NK cells are always active. An interaction with normal, intact MHCI molecules on a healthy cell disables the killing sequence, and the NK cell moves on. After the NK cell detects an infected or tumor cell, its cytoplasm secretes granules comprised of perforin, a destructive protein that creates a pore in the target cell. Granzymes are released along with the perforin in the immunological synapse. A granzyme is a protease that digests cellular proteins and induces the target cell to undergo programmed cell death, or apoptosis. Phagocytic cells then digest the cell debris left behind. NK cells are constantly patrolling the body and are an effective mechanism for controlling potential infections and preventing cancer progression. 1198 Chapter 42. The Immune System Access for free at OpenStax.org. Complement. An array of approximately 20 types of soluble proteins, called a complement system, functions to destroy extracellular pathogens. Cells of the liver and macrophages synthesize complement proteins continuously. These proteins are abundant in the blood serum and are capable of responding immediately to infecting microorganisms. The complement system is so named because it is complementary to the antibody response of the adaptive immune system. Complement proteins bind to the surfaces of microorganisms and are particularly attracted to pathogens that are already bound by antibodies. Binding of complement proteins occurs in a specific and highly regulated sequence, with each successive protein being activated by cleavage and/or structural changes induced upon binding of the preceding proteins. After the first few complement proteins bind, a cascade of sequential binding events follows in which the pathogen rapidly becomes coated in complement proteins. Complement proteins perform several functions. The proteins serve as a marker to indicate the presence of a pathogen to phagocytic cells, such as macrophages and B cells, and enhance engulfment. This process is called opsonization. Certain complement proteins can combine to form attack complexes that open pores in microbial cell membranes. These structures destroy pathogens by causing their contents to leak, as illustrated in Figure 42.7. 42.1. Innate immune response. 1199 Figure 42.7. The classic pathway for the complement cascade involves the attachment of several initial complement proteins to an antibody-bound pathogen followed by rapid activation and binding of many more complement proteins and the creation of destructive pores in the microbial cell envelope and cell wall. The alternate pathway does not involve antibody activation. Rather, C3 convertase spontaneously breaks down C3. Endogenous regulatory proteins prevent the complement complex from binding to host cells. Pathogens lacking these regulatory proteins are lysed. Credit. Modification of work by NIH.